So good evening, everybody. Thank you for joining us this evening for our latest alumni digital event. This is about our fifth or sixth event now in this format, and we're really pleased that so many of you have registered to join us this evening as the sun pokes through the rain clouds just as we come to the end of today. Do hope you're all well and are healthy and fit and enjoying the start of a new academic year as we are in Bristol. So for those of you who I've not met before, my name is Andrew Monk and I have the privilege of being the Director of Development and Alumni Relations at the University of Bristol. So this evening's event, we're very grateful to Professor Matt Jones, who is joining us this evening to talk about the science of sleep, a topic I've heard him talk about a couple of times before, but is endlessly fascinating. And I think everybody should just be grateful that I chose not to wear my pajamas for this talk, although hopefully we can stay awake till the end and then get a good night's sleep on the back of it. So from me at this point in time, there are just a few housekeeping details that I just wanted to share. So you'll see on the bottom of the screen, you've got an opportunity to ask questions, a Q&A tab. Please do use that to ask any questions you'd like to. What the format that we'll run is that Matt's got about a half hour or so presentation. We've then got a few questions that were submitted in advance, and then we'll go to the live question board and answer as many as we can before we get to about 8 o'clock or just after. So use the question and answer tab for any questions that you have. If you've got any technical issues at all or any issues with the stream at all, then please email alumni events@bristol.ac.uk and they will help you resolve any issues. We often say log out and log back in, and that solves an awful lot of issues along the way. For those who are less familiar with Zoom as well, just so that you're aware, there are options that you can set around the display settings for your screen. So you can shrink or increase the size of the slides once Matt starts sharing those. And indeed, you can hide speakers who haven't got their camera turned on, just so that you can really see everything that's going on and see the information that we're able to share. So I think that's all of the housekeeping details that we have to share at the moment. And so on to the main event, really. So ever so grateful to Matt for giving up his evening and for missing bedtime with his children this evening, so he can share with us a little bit more about science for sleep. So without further ado, I will hand over to Matt.
Thanks very much, Andrew. Let me just fiddle around momentarily while I get my slides up. Okay, good evening, everyone. Thanks so much for joining me. It's a great pleasure to welcome you here to the Jones family study, which is also the Jones family music room, occasionally the Jones family home classroom, Pilates studio, and now lecture theatre as well. So we're lucky to have such a versatile room. As Andrew said, my name is Matt, and I'm a neuroscientist, and in these exasperating times, I sometimes pause to wonder how that came about. So I'll quickly share that story with you. Like many of you, I presume, my career has been bolstered by the University of Bristol. But unlike the majority of you, I think, I owe my very life to the University of Bristol in the sense that my parents actually met here as undergraduate students. And well, one of the unfortunate consequences of that meeting is me. So my mum studied botany and zoology here in Bristol, and my dad was studying M.Ed, who specialised in Shakespeare in English literature. And like all eager to please adolescents, I really wanted to honour both my parents' legacies, and so I took the average of the fascinating biology that mum worked on and the poetry that dad loved so much, and that led me to the brain, which I felt offered the perfect combination of those two facets. So there you go, a straightforward but life-changing story that led me to work here in Bristol as a neuroscientist. So when I was made director of Bristol Neuroscience a couple of years ago, I wanted to maintain a straightforward but life-changing story. And that's why I led our strategic focus around these four cornerstones of all our lives. So the four research hubs of Bristol Neuroscience focus on mental health, on memory, on movement, and on sleep. Now, of course, All these four cornerstones of our lives have been constrained during the current pandemic. And this evening, as Andrew said, and as you know, I'm going to focus on the latter, on sleep in particular. I will touch upon the impact of COVID-19 and the pandemic on sleep to some extent, but I don't want to dwell on it too much. I'll be happy to answer more questions though. Now, sleep has been enjoying the headlines to an extent for several years. It certainly graces the health and lifestyle pages of the broadsheets on a regular basis. And that's normally good news, but it does risk becoming a bit faddish at times. There's only so many times you can read an article suggesting that you don't drink caffeine, don't drink alcohol, buy thicker curtains, and so on, to improve the quality of your sleep and thereby your life. And I'm not going to preach to you about sleep hygiene this evening, although again, I'm happy to answer questions about it at the end. Rather, what I want to focus on is how researching sleep, how understanding what's happening in the brain during sleep really fuels an opportunity to harness sleep to our best advantage and indeed, therefore, to improve the quality of our lives. And that relates to the overall mission of Bristol Neuroscience stated here. Our mission is to do brain research for better lives. Now, before I dive into that brain research, we just need to catch up together, a quick Sleep 101 primer, though I'm sure most of you are familiar with the basics, since you spend about a third of your own lives asleep. And in fact, every animal that's been studied to date does appear to sleep. Now, in mammals, and again, all mammals that have been studied to date, sleep can be subdivided into two broad stages. The best known is probably REM sleep, or rapid eye movement sleep. But REM is partnered by non-REM sleep, a rather disparaging title, non-REM, or sometimes called slow wave sleep. And in humans, non-REM sleep is subdivided further into stages 1, 2, and 3. Now, during the course of your average night, you get about 8 hours of sleep, shown on this graph here, and you cycle repeatedly through a phase of non-REM to a spike of REM, another phase of non-REM, another spike of REM, and so on. The proportion of non-REM sleep is higher at the start of the night. That's when you have your deepest sleep. And the proportion of REM sleep is higher towards the morning when you're waking up. And if you wake up out of REM, as one should, then you feel wonderfully warm and relaxed. And in part, that's because all your muscles completely turn off during REM. You don't perform any body movements normally during REM sleep. Now, one of the greatest mysteries to me, and in fact, one of the subjects of my main grants in the lab, is why do we do this alternating cyclical rhythm throughout the course of the night? It's hard enough to ascertain precisely what these different stages of sleep, what functions they subserve, but we really have no idea why it is that we cycle in this way, alternating between non-REM and REM. To me, it kind of whiffs of some sort of algorithm whereby something changes in the brain during non-REM sleep, then perhaps we check whether it's changed enough during REM sleep, then go back to non-REM, do a bit more changing, back to REM again to check, something like that. But it's really, there's no firm theory about why sleep architecture occurs in this way. So there are many mysteries yet to solve in this research domain. Now, one way we classify these sleep stages is using electroencephalography, or EEG, ably demonstrated here by Ulrich, one of my research team, who's sporting some electrodes which are glued onto his scalp, rather fetchingly. And he's about to settle down for a night's sleep. During that night of sleep, we'll record his brain activity, his brain waves, through these EEG electrodes glued to his scalp. We'll also record his breathing, his heart rate, his muscle movements, and so on. That whole suite of recording is known as polysomnography. In fact, Ulrich is about to settle down in Bristol's Clinical Research and Imaging Centre on St Michael's Hill. Though sadly, one ramification of COVID is that imaging centre is being shut down by the university because we can't afford it anymore. COVID, as I'm sure you're aware, has induced financial panic amongst many other types of panic. So the brainwaves that we record during non-REM and REM sleep look something like this, a whole bunch of wiggly lines, which neuroscientists like me love. Up at the top here, we've got the EEG activity, the brainwaves that are evident during wakefulness. Down at the bottom, we've got the brain waves that are evident during REM sleep. And you'll see the two look rather similar. And in fact, REM sleep is sometimes called paradoxical sleep, because paradoxically, the brain activity that you record from the surface of the brain during REM sleep looks rather similar to the activity during wakefulness. And then the stages 1, 2, and 3 of non-REM sleep look quite different. The deepest sleep, or stage 3 non-REM sleep, has these slow waves, so these big cyclical oscillations about once every second, which kind of act as a metronome timing brain activity during those stages of deep sleep. Now, EEG recording like this has a proud history in Bristol, as it happens. Some of the early EEG recordings were developed here in the context of the Bristol Neurological Institute, which at one point was housed in that big yellow building you can see from the M32, should you ever have the misfortune to have to navigate the M32 during the rush hour. Now, it turns out that your own personal pattern of sleep brainwaves is highly characteristic of you and is highly related to your genetic makeup, to your genes coded in your DNA. The patterns of brain activity that occur during sleep can be represented in these graphs. And essentially, this is summarizing the types and amplitudes and rhythms of brainwaves that an individual shows during their non-REM sleep. Now, in this graph, there are two lines. There's a greenish line and a bluish line, same for all four of these panels. And those different colored lines represent the patterns of brain activity from two different individuals. But in this top left-hand graph here, the two individuals are genetically identical. So they're monozygotic identical twins. And you see that the pattern of brain activity during non-REM sleep is also almost identical across these two twins. Whereas if you take unrelated females, in this case, of the same age, about 11 years of age, their patterns of brain activity during non-REM sleep look very different. So again, there's this fingerprint of you that we can assay through the EEG activity during non-REM sleep that reflects your genetic architecture. That's incredibly exciting and powerful, I think, as a means to understand our individual biologies. This pattern of brain activity also depends on age. So you'll see as you transition from 11 years old to 50 years old, in this case, then the fingerprint of brain activity during non-REM sleep also changes. So your own individual pattern does reflect you, but it also reflects the time of life that you're at. So that presents lots of really exciting opportunities for research, mapping changes in the individual's brain activity over the life course. And I'll touch upon some of the examples of how we're using that kind of approach in Bristol later in the talk. So to understand sleep, you need to study brain activity during sleep at many different levels. And that's the integrative approach that we take here in Bristol Neuroscience. So in some cases, that means we need to understand at the cellular and molecular level what's going on in the brain. And that's very hard to do in humans. And so we would turn to animal models. So here's a rat curled up asleep in the lab. And working with rats allows us to manipulate their genome, for example, to record the activity of individual neurons in their brains. But of course, always has its limitations because rats aren't humans. So we turn to volunteers. Here's another volunteer sporting our EEG recording net. Some of these volunteers are healthy, whereas others are suffering from a range of disorders which are associated with sleep abnormalities. We use a range of devices to record brain activity during sleep. So as well as these kind of full-blown clinical devices shown in the center of the slide, we also have these commercial personal devices shown in this headband here, which has fewer EEG electrodes, but no wires, so it's very easy and comfortable to wear for participants during the night. We may also sneak into your home, with your consent, I hasten to add, and instrument your house with a whole bunch of sensors measuring your movement, measuring environmental conditions, videoing your behavior, and coupling all those measures to measures of brain activity in the form of EEG, which allows us again to get this really deep personal fingerprint of how your biology is reflected in your daily living and vice versa. We might instrument you with some other devices. So this U-Rhythm device was developed by Stafford Lightman and his team here in Bristol. And that allows continuous measurement of hormone levels from blood samples, for example. So by coupling all these different measures together, we can get a very detailed fingerprint of an individual and a very sensitive measure of how disease, for example, might be changing that individual for the better or for the worse. So before I talk in a bit more depth about some Bristol examples, let's visit the boudoir of fame, as I've called it, some notorious sleepers from across the animal kingdom, which shed some light on what sleep might be useful for. So first up, can anyone guess what first up is going to be? Yes, that's right. I'm sure many of you guessed. First up is the famous jellyfish, which only a couple of years ago, it turned out, does appear to sleep. So monitoring jellyfish in California, some researchers at Stanford noticed that during the night, the jellyfish stopped moving as much, and they appeared to sink deeper down in the water. They stopped eating and appeared to enter a sleep-like state, despite the fact that the humble jellyfish doesn't have a brain. It does have a nervous system, but it doesn't have a kind of central hub of the nervous system in the form of a brain. So it seems that sleep is a very primitive, primordial process that percolates across the animal kingdom. Next up, we have a very, very cute duck-billed platypus curled up asleep here. Now, they're mammals, of course, the duck-billed platypus, but they're monotremes, so they branched off from our evolutionary tree a very long time ago. They're sort of primitive mammals. It's very hard to study sleep in the duck-billed platypus because they're extremely sensitive to their electrical environment. So the bill of the duck-billed platypus is an electrical sensor which is bombarded by standard laboratory equipment. And in fact, many platypus die within a month of being caught and held in captivity because of all this electrical noise. So it took a while for people to figure out that they had to do their experiments within a metal cage, basically, which shielded the platypus from all this interference. And under those conditions, when the platypus sleeps naturally, it has almost all REM sleep. So it doesn't cycle from non-REM to REM sleep like placental mammals like us, for example. And it's not really clear at all why that is or what that means for the platypus. How is it that an animal has almost all REM sleep, can apparently perform all the same sleep-dependent functions as mammals that don't? One of my personal favorites here is the fur seal. Now, the fur seal, like the dolphin, when it's at sea, can sleep half a brain at a time. So it stays alert enough to prevent itself from drowning or being eaten by sleeping one hemisphere at a time. An enviable skill, I'm sure you would agree. But when the fur seal returns to land, and then it just sleeps like many other mammals, sleeps in the same kind of way as a dog would, for example. And the proportion of non-REM and REM sleep that seal shows, again, is dependent on whether it's at sea or on land. And has prompted some suggestions that actually the function of REM sleep might relate to temperature regulation as much as anything else. Again, it's amazing to me how little we know about what sleep is for, given how much of our lives we spend asleep. And well, that's one good reason to be a neuroscientist, although it's a bit embarrassing that we haven't made more progress sometimes. Now, this final figure here shows a human family and the inheritance of a particular gene mutation. The gene is shown down there, catchily named ADRB1. And this family passed to one another the ability to not require much sleep. So this is a family who on average sleep for 4 hours per night and perform perfectly well with that short amount of sleep, feel perfectly healthy and are very happy because they can achieve so much more with or have so much more waking time than the average human. So we're beginning to understand more about how it is that these non-sleep requiring folk come about. As this demonstrates, it relates to very rare mutations in particular genes. So this particular mutation only occurs in about 40 of each 100,000 people. So it is very rare. But the fact that the folk can get by on much less sleep is really fascinating. And this was only discovered a couple of years ago. More studies recording their brain activity and understanding their body systems in greater detail may eventually lead to a situation where many more of us don't need so much sleep. Whether or not we'd want it is another matter, of course. Now, there are other altered states of consciousness, of course, some of which appear very appealing to me personally, and one of them is hibernation. Now, hibernation is not the same as sleep. In fact, a hibernating bear, shown up here, for example, does wake up periodically from hibernation in order to go to sleep. So again, that evidences how important sleep is. But still, hibernation is a sleep-related state, an altered state of consciousness. And it's very common in many mammals, actually. There's also a similar state called torpor, which is essentially a brief hibernation. The body systems sort of shut down. The animal, in some cases, even primates go into torpor. The animal sort of rejects life temporarily and then reawakens to appear quite normal and unaffected by this state of torpor. Now, for this reason, torpor caught the eye of one of my PhD students, Dr. Mike Ambler, working with my colleague, Tony Pickering. Now, Mike is an intensive care clinician, and as such, spends a lot of his time trying to make sure that brains stay healthy in intensive care. And typically that means making sure they're well supplied with oxygen and food and so on, keeping the metabolism of the brain on track. But that's quite different from the metabolic state of a brain in torpor, in a mouse, for example, which goes into torpor quite naturally. So Mike began to wonder if he could induce torpor in a species that wouldn't normally show torpor, and thereby harness the neuroprotective effects of this state, eventually perhaps using this to inform his intensive care work. So I'll briefly summarize Mike's approach here. This graph shows data from a single laboratory mouse, and lab mice can be encouraged to enter a torpor state if you chill the room that they're held in down below 20 degrees, and you restrict their access to food. So under these conditions where their metabolism is challenged because they have to stay warm and they don't have enough food to eat, then they enter a torpor state, shown on day 4 here of this red trace, for example. You see the mouse is becoming torpid for a few hours, but it recovers and is up and running again perfectly happily. So Mike's plan was to capture the neurons in the brain that control entry into this torpor state, and then harness them to control entry and exit from torpor. And he did this by making a super mouse. This super mouse, shown in the middle here, allows us to control particular neurons in the brain that are activated when the mouse goes into torpor. So we express in those neurons a protein whose activity can be controlled by a drug, which is called 4-OHT. And that means that whenever we inject the drug into the mouse subsequently, we can induce a kind of synthetic torpor. And that's what's shown on this second graph of Mike's. So here a mouse is kept warm, is well fed, but we inject a drug here called CNO, which activates the parts of the brain that would normally trigger the mouse to go into torpor. And you see that the drug induces this torpor-like state. And during this state, the brain shuts down to a large extent and yet remains very healthy and fully functional on awakening. So it's a slightly science fiction experiment. And when Mike first approached me to suggest it, I tried to dismiss it as just far too bonkers. It's never going to work. And thankfully, his talent and persistence has shown me wrong. So the idea now is, okay, now we can control torpor in a mouse because we know which parts of the brain trigger that state. Perhaps we could induce torpor in a rat, for example, which wouldn't normally enter that state. And in the longer run, perhaps we can understand the brain circuits that control torpor well enough to replicate some aspects of this state in intensive care and thereby protect the brains of patients. We're also doing a lot of work with the cardiac surgeons in Bristol, Dr. Ben Gibbons, for example, who are interested in the quality of sleep in intensive care units following cardiac surgery. So we're introducing some of these methods to monitor sleep in great detail into the intensive care unit to understand how sleep quality can affect recovery from major operations like heart surgery, for example. Now, one of my own interests in sleep relates to its support of memory processing. And that's fascinating in relation to hibernation as well. The squirrel hibernates, wakes up, and knows exactly where in your lawn it buried its hazelnuts. How on earth is that possible? And sleep's contributions to memory have been recognized for a good couple of thousand years by this very Roman-looking Roman chap, for example, who was paraphrasing what we say nowadays, that, you know, just sleep on it and it will make sense in the morning. Why is that? So what is it that's happening during sleep that supports that kind of mechanism? Well, here's an experimental example of sleep's contributions to memory processing, to our making sense of our waking lives. In this experiment, participants come into the lab and we train them to type a sequence of 4 numbers as quickly and accurately as possible, just on a computer keyboard. So over 12 training trials, they get better at typing the correct sequence. We then give them a night's sleep again in the lab whilst we're recording brain activity, and test them again the following morning. And in healthy participants, shown in blue here, they perform better the following morning than they did at the end of training on the previous day. Whereas in this case, in schizophrenia patients, shown in red here, they don't show this overnight memory improvement. And in fact, we can predict how well or badly individuals will do in this memory task the next morning on the basis of their brain activity during the intervening night. This is just one example of sleep's contributions to memory processing. Which parts of the brain are key to this? Well, a central hub of memory processing is a brain region known as the hippocampus, shown up here. How do we know this? Well, for example, if you grabbed a London cabbie and you shoved him or her in a brain imaging scanner and asked him or her to recall their route around London town, and then looked in the brain to see which part of the brain was most active as that memory is recalled, this is what you'd see, this yellow hotspot looking top down on the brains here. So the nose of participants is up at the top, the back of the head is down at the bottom. This hotspot is the right hippocampus, which is a central hub for memory processing. Now at a cellular level, here's what's going on at the hippocampus. As you move through space, as you navigate your way around the world, individual brain cells or neurons in the hippocampus are active in different locations. These cells are called place cells. So as a rat, for example, follows this dotted black line across the screen, different place cells are active in different locations, and collectively, they can be used to encode the location of that rat. It's mind-blowing stuff to me. I mean, how do you know where you are? I'd never really thought about that. You know where you are because some specific subset of your 80 billion neurons is selectively active in your current location. If you move slightly, then a different subset of your 80 billion neurons is active in that location. That's the basis of your cognitive map. And these place cells were discovered by John O'Keefe, working with his colleague John Dostrowski back in 1971. And related neurons called grid cells were discovered by the Moses, who work in Norway. And collectively, this trio won the Nobel Prize in 2014. I show this slide partly to show off, because I've had the honor of working with all these Nobel laureates, but also to forewarn you that winning the Nobel Prize can be a dangerous business. These photos were the original publicity photos, but they were updated on the Nobel website. I'm not sure why. So these very dark and brooding photos. John, for example, is a lovely guy with the most infectious laugh I know, but he looks downright evil in this updated photo. So I took this as evidence that I really shouldn't bother winning the Nobel Prize. I can sadly add that the husband and wife team of May-Britt and Edvard also divorced after they got the prize. So it's a dangerous business. Anyway, back to the hippocampus and to sleep. If this hippocampal structure is so important for memory, if it contains your cognitive map that stores your memories of the world, what's happening in the hippocampus during sleep? Well, that's what this slide shows. Here's the average activity of the hippocampus in a rat that's asleep for the first hour of the experiment, then running around on a maze, learning where to find chocolate for the second hour of the experiment, then back asleep again. And you'll see that in this final hour of the experiment, when the rat is back asleep, the hippocampal activity is actually at a higher level than it is when it's learning about its world. So what on earth is going on here? Well, to summarize in cartoon form, as the animal is making new memories, then patterns of place cell activity in its hippocampus form those memories. But when it subsequently goes to sleep, those patterns are replayed, they're rehearsed multiple times in this unconscious sleep state. And that process of replay consolidates the memory that the animal has just formed. Now this mechanism was first uncovered by Matt Wilson, working at the Massachusetts Institute of Technology. And Matt was my boss when I was a research fellow at MIT prior to coming to Bristol. And so this is a good opportunity to thank Matt for introducing me to the joys of the sleep state, I guess. Now, one of my other favorite neuroscientists is this chap, Karim Bonchanan, and Karim works in Paris. That's in Europe, by the way. And as you might tell from the photo, Karim is a lovely guy to drink wine with, one of my favorites. And he did a really cool experiment. So he was recording these place cells from the hippocampus of rats. These are 5 different place cells shown here. What we're looking at in these hotspots is the region of a circular arena, about 1 meter in diameter, where an individual place cell is active. So these 5 different place cells signal 5 different locations within this 1 meter diameter environment. Having established that, Karim then lets the rats go to sleep. But every time this individual place cell at the top here fires some activity, then Karim stimulates parts of the brain which is the reward signaling pathway. So without waking the rat up, he couples the activity of the single brain cell with a reward signal. The rat is completely oblivious to this because it's still asleep. What we're looking at here is the behavioral activity of the animals. So before this experiment, the rats mooched around at random in the circular arena, so there's no particular hotspot of where they like to spend their time. But after the activity of these different place cells was paired with the reward signal, then the rats couldn't help the next day but gravitate towards where those neurons are active. So what Karim has shown here is that by pairing brain activity during sleep with a reward signal, without affecting consciousness, without waking the animal up, you can control the activity of the animal on the subsequent day. Now, could this be applicable to the human condition? Well, that sort of cellular resolution experiment is rarely feasible, but we can control brain activity by applying electrodes to the scalp and delivering carefully controlled amounts of current. So this experiment done by Jan Born in Tübingen in Germany showed a proof of principle here. People learned a list of words. They then went to sleep, and they either received an electrode setup with no meaningful stimulation, or they received an electrode setup with stimulation that mimicked this slow wave activity that occurs during non-REM sleep. So this 1 cycle per second oscillation that hallmarks our deep sleep. They were then tested in the word list the next morning. And if they received the slow wave stimulation shown in this black bar here, they remembered twice as much as people who didn't receive the stimulation. So this opens the door to a range of opportunities to modulating brain activity during sleep in order to control or finesse or cure aberrant behavior. There are other ways of modulating brain activity during sleep, it turns out. Any of the audience who've had children may have spent some time rocking babies. I know both my kids were rocked like crazy in their early months. And it turns out that rocking at about once per second, as long as that rocking coincides with the right phase of sleep, so this deep non-REM sleep, actually improves the quality in neuroscientific terms of that sleep. Now, this has been shown in laboratory mice, but it's also been shown in humans. I personally love to sleep in a hammock. It doesn't get the opportunity very often, but now I know why. And if that hammock is rocking at the right frequency and at the right time during my sleep cycle, it really can augment the quality of my sleep. There are some drugs as well that can augment sleep quality. And these Bristol researchers, Liz Coulthard, who you may recognize, and Hannah Isatalis, who was a PhD student with Liz at the time and now a lecturer in digital health in Bristol, had the idea that L-DOPA, which is a very safe, commonly used drug, used in the treatment of Parkinson's disease because it increases the levels of the neurotransmitter dopamine in the brain. They thought that giving L-DOPA to healthy participants after learning and just before they went to sleep might actually improve the quality of that sleep and thereby improve the quality of the memory. And so in this trial known as Dopamind, that's exactly what Liz and Hannah have shown. These graphs show that the L-DOPA drug increases the amount of this rhythmic activity that occurs during sleep. And it also enabled participants to remember more accurately what they'd been trained on prior to receiving the drug. So this is an exciting opportunity to use a cheap, widely available drug to augment sleep quality in those where it's compromised. Of course, as I said, L-DOPA is used already to treat Parkinson's, and this relates to another study we're involved in, led by Amber, who's a fantastic PhD student in my lab, who sleeps wherever she can, as demonstrated by this inflatable hammock job on our lab retreats last summer. Now, Amber is working on REM sleep behavioral disorder alongside two neurologists, Harry and Alan, based up at Southmead Hospital, Raoul, who's a machine learning computer scientist, and Keith, who's the director of Next Generation Therapeutics at a pharmaceutical company. We collaborate with Eli Lilly. Now, REM sleep behavioral disorder is shown in this video here. As I mentioned earlier, normally during REM sleep, we're still, our muscles stop working. But in this patient with RBD, in REM sleep, he starts to move around. In fact, he's thrashing around and he's acting out a violent dream. So he starts beating the bed at this point. And that's quite common in RBD. Patients lose their muscle atonia during REM. They start moving during REM and acting out dreams, which are often quite violent. So it often comes to the clinician's attention when patients beat their bed partners. Now, the horrible thing, if that wasn't horrible enough, is that 12 years after diagnosis with RBD, you've got about a 75% chance of getting either Parkinson's or multiple system atrophy or Lewy body dementia. None of these are things that you want. But at this point, we have no way of predicting which degenerative disorder you might progress onto or how far down that progression you are. So Amber is recording sleep EEG in REM sleep behavioral disorder patients, applying the machine learning analysis under the watchful eye of Raoul, and asking if we can detect very early on and predict which of these horrible paths patients might be on in order to intervene at an earlier stage. Now, I said I'd talk briefly about sleep in the pandemic. And again, it's Liz Coulthard, now without the mask, who's led the charge here. Liz set up a Sleep Quest, an online survey during April to May of this year, during the lockdown, to ascertain what effect it was having on sleep quality. And there was a number of confirmatory results. On average, people's sleep quality had deteriorated during lockdown. The thing that surprised me most was that older people's sleep quality was less affected than younger people. Now, we don't know why that is yet, but it came as a surprise to me. Whether it relates to social anxiety or financial insecurity, we just don't know yet. But it rings alarm bells for me, given how important sleep is throughout the life course. Sleep's important in babies, it's important in adolescents, and it's important and impaired in old age. I'll skip over this in the name of time. So we are doing kind of cradle to grave work in Bristol. John Evans is a senior lecturer in mental health who's interested in particular in postnatal depression. And he has some evidence that bright light therapy, so using these glasses to deliver bright light to people and then train their sleep patterns during postnatal depression, can actually significantly improve the effectiveness as antidepressants and make their recovery quicker. So that's one of the projects that we're just getting off the ground at the moment. We're also very concerned about adolescent sleep, which gets a bad name for itself. The stereotypical adolescent is portrayed as a lazy bum who won't get out of bed. But as many of you will know, that's because their biology is shifting. So their biology is telling them to get out of bed later and to go to bed later as well. So by insisting they wake up early, we're actually constraining the amount of sleep that they're able to get. And this could have really important ramifications across the board because the pattern of brain activity during sleep is undergoing really sensitive changes that reflect brain development during adolescence. And that's shown here. Here we're looking at the patterns of brain activity during sleep. So again, a top-down view of the head, the nose at the top, back of the head down at the bottom, ears on either side. We're comparing between ages 2 to 5 on the left and 17 to 20 on the right. And you see there are massive changes in the structure of brain activity during sleep across this time span that reflect changes in brain networks during adolescence. So if we disrupt adolescent sleep, we stand a dangerous chance of interfering with adolescent brain development. So that's prompted one of the projects that is growing out of Bristol's Sleep Hub right now, led very ably by Professor Esther Crawley, who's the director of our Centre for Child and Adolescent Health. Esther's plan is to use smartphones to monitor adolescent sleep, to ring alarm bells early if somebody's sleep patterns change in a in a detrimental way. And then to also use the smartphones to deliver an intervention, so to deliver internet cognitive behavioral therapy to improve sleep quality before it's too late, essentially. So to intervene before the rot has set in and before the lasting damage to brain development has been done by sleep disruption during adolescence. So, I, along with many other people, are really concerned about the impact that the pandemic and many other unfortunate societal changes are having on adolescent brain development, because if we don't deal with it now, then we're going to be paying the price for many years to come. So I'll wrap up there and look forward to answering some of your questions. As I said, the mission for Bristol Neuroscience is to do brain research for better lives. And I think that necessarily has to include researching sleep. Sleep is never the whole answer. It's always part of the story. It interacts with the rich tapestry of our lives. Sometimes bad sleep reflects other things that are going on. Sometimes bad sleep causes other things to go on. But it really is a central part of the human condition, which neuroscience still has a lot to contribute to, I think. Thanks very much. And I think Andrew is going to field some questions.
Thank you ever so much, Matt. Absolutely fascinating as ever. And we have a range of questions. So I've got a few that were submitted beforehand, and then we'll go on some of the live questions as well. So one of the questions that we had before was from Louisa, who said, what are the links, if any, between sleep loss, psychological trauma, and the development of nerve damage? So, for example, MS or Parkinson's.
Well, thanks to all of you who very conscientiously submitted questions beforehand. You're far more organized or conscientious than I ever was. That's a really important question that relates to quite a lot of the Parkinson's work that's going on in Bristol and other places. The short answer is there are lots of associations. Sleep loss is never a good thing, and it clearly exacerbates immune system dysfunction and inflammation, both of which are early antecedents of pathologies like MS and Parkinson's, which involve frank inflammation. But at this point, the field is kind of stuck with the association and isn't yet clear whether preventing the sleep loss early on would be enough to either slow progression or to cure the diseases. That's why we're at a stage where we're monitoring sleep carefully in large populations of patients and trying to better pin down those mechanistic pathways. But it's an exciting time because we know those associations are there, but kind of a disappointing time as well because it hasn't got beyond that association stage yet.
Thank you ever so much. And sorry, just one point that I did mean to mention at the beginning, just there have been a couple of questions coming through. This whole lecture and this whole session is recorded, and we will share a recording of this with everybody who's here this evening, everybody who's registered for the event, and with wider alumni that you can share and rewatch at any time. So in case anybody wants to share this further or revisit anything that's been discussed, then you will have that chance at a later point as well. So another question that came in was around, so from Anu, was around sleep, and you've talked about the effect of sleep and memory and how it improves our memory. The question is, how can sleep help us to make better decisions? So the talking about things about I'll sleep on it and then arrive at a decision tomorrow. Is there any scientific basis in that?
Another great question, and yes, there is. I guess there are kind of two levels that sleep contributes here. One is that poor sleep leaves you predisposed to impulsive and unethical behavior. There's quite clear epidemiological evidence for that. So it can corrupt your decisions in that way. Good sleep supports good decision making through its effects on memory, really, because your decisions are made on the basis of your model of the world. And the central function of the brain is to create your model of the world and try and predict what you think you ought to do in order to optimize what happens next. So through its support of memory consolidation, through its selective strengthening of useful memories and selective weakening of the detritus, the noise, if you like, then sleep can support decision-making in that way. And I mentioned that the replay of patterns of neural activity that occurs during sleep. It's not the case that every pattern of neural activity that occurred during wakefulness is replayed. Rather, there's preferential replay and strengthening of salient or important patterns. So for example, the route that you took to find your unexpected chocolate bar is the route that you will replay more, as opposed to the route that led to a dead end. So by tuning memory in that way, sleep supports, helps to optimize decision making subsequently.
I'm going to go and find one of those routes that leads to a chocolate bar.
It has to be unexpected, Andrew. Otherwise,
no, it has to be unexpected. Okay, I'll work on that certainly. A question from Ian, combining history and sleep. So do we have any ways of estimating how much our ancestors slept in previous millennia?
Yeah, really interesting. To an extent, yes, although the trouble with history is it's always to an extent. So there's a guy at UCLA called Jerome Siegel who's done a lot of work in this regard. He's the same guy who recorded sleep in the platypus, by the way. So he's been studying hunter-gatherers whose lifestyle, so far as we know, is much more reminiscent of the lifestyle of our ancestors. And he studied 3 different tribes, and I think in South America and Africa. And there were a couple of surprises. So they, on average, they sleep for about 6.5 hours per night. So that's actually less than the average in the developed world. They didn't nap. They very rarely took a nap. But the levels of insomnia were negligible. There was essentially none. So there's lots of things we're doing wrong in modern society, most likely, particularly or in relation to sleep, that are perhaps pulling our biology away from its innate state. But there are some surprises. Perhaps if we were sleeping properly, then 6.5 hours would do, and quite what it is that means we're not sleeping properly. There's lots of parameters in that equation. But it's very powerful to look back at these other ways of life and think how they're affecting sleep. It wasn't, the bedroom only came to be quite recently, right? We used to sleep with our animals in the barn, and then the wealthy were able to creep up to the hayloft or something, and then beds appeared. So we have the rate of change of our approach to sleep has been quite dramatic relative to the time course of humanity as a whole. So I'm all for going back to hunter-gathering. I'll see you on the downs, Andrew.
Jolly good. So a question that's come in from Diana, which is relevant and causes much debate in our house, as I am very much a night owl and my wife is very much an early riser. So do sleeping patterns, night owls versus early risers, matter for productivity? How easy is it to switch from one to the other?
So you could argue that sleeping patterns don't matter for productivity, but waking patterns relative to sleeping patterns do, right? So the sleeping pattern isn't to blame if it happens to be different. What's to blame is society trying to force you to align your waking life to everyone else's. So there's no reason to think that an owl versus a lark is more or less productive, but productivity does vary because it's contingent upon how your sleeping pattern aligns with the norm as it stands at the moment. How easy is it to change?
Yeah, I was just about to say, how does it work if we are then forced, if you're naturally aligned to being a night owl, but societally you're forced into a situation where you have to be a lark?
Yeah, so I think I'm sure there are different reasons for being owls or larks, but in the case where your genome, which always gets the blame, is the primary driver of your sleep timing, then it's not easy to change, and it's unreasonable to expect people to change. And a sort of temporary example of that relates to adolescence, where we expect them to align at a time when their biology just doesn't. Assuming that you're driven that way, then it's not easy to change, but it's not easy to fit in if you don't.
And I mean, interestingly, you mentioned adolescence, because on the live questions that came in, I'm just scrolling through to try and find it now. There are a lot on here, but I'm sure I read one which was asking about, are there any tips on how you help adolescents who have settled into a sleeping pattern in lockdown and are now returning to school, and so we're forcing them to change that sleeping pattern. Is there anything that can be done to support them or help them in terms of adapting that sleeping pattern that's allowed to fall into that natural rhythm?
Well, so I think this relates to one of the other questions that was submitted beforehand, perhaps, about the timing of the school day. And there has been a study in Seattle. The paper is called Sleep More in Seattle, where they started the high school day later. And that was shown to improve the grades of pupils, not hugely, by about 5%, but also improve attendance. So, were we to act as a society, we could perhaps help adolescents by changing the timing of the school day, assuming we're run by a government that couldn't organize a public health piss up in a brewery. That's not going to happen anytime soon. So in the meantime, don't nap. Like the hunter-gatherers, napping tends to confuse things. Don't get angry about it. It's just biology, Mum. There's nothing they can do. Be sensible about the lighting and the caffeine and everything else. The blue light filters on the phones do some good. My household bans looking at phones after 9 o'clock, which is probably later than it should be, but and banning things from adolescents generally doesn't go down very well. But talk about the biology. I can send you the links to the papers if you like. Softly, I think. I mean, to shift the timing of your sleep does take time. So if you're jet lagged, for example, it takes about 24 hours to recover from each hour of jet lag. And so you could think of this as a social jet lag and approach it in that way and figure out how long it should take for them to realign accordingly. I urge scientifically informed patience.
And so one of the questions that come through live from Hayley is that so she is asking, as she is neurodiverse herself and identifies as such, and she seems to only function on 9 to 10 hours sleep a night. Is there a link between neurodiversity and the need for sleep?
Again, yes, there is a link. There's a statistical association, but it is a very diverse link. There's no straight line from a particular sleep pattern to a particular form of neurodiversity. It's taken a while for people to accept that different sleep patterns might be an important part of neurodiverse phenotypes. They were often dismissed by psychiatrists, for example, as just an effect of the drugs or whatever. Again, it's only in the last few years that we've started to uncover more specific links between sleep patterns and just different people, just different individuals. So it'd be really interesting to see, Hayley, of the more sleep that you need, how much of it is REM, how much of it is non-REM, how is the sleep architecture, for example. Maybe there's some aspect of sleep quality as opposed to sleep quantity that could be worked on, such that you didn't need to sleep for 9 to 10 hours anymore. And I'm sure that's true for many of us, I think. People, it's easy to confuse sleep quantity with sleep quality, and they're just not the same thing. That's why it's so important to understand what's going on in the brain.
So on the topic of sleep quality, a question from Hayley is, does the use of alarm clocks affect our hippocampus?
I don't know, and I'll resist making stuff up. It's clear that your brain activity aligns to regularly timed events. So that includes alarm clocks, but it also includes eating, for example. So the timing of your sleep is strongly controlled by your circadian rhythm. And one of the strongest entrainers of your circadian rhythm is the time at which you eat. Light is the other famous entrainer of your circadian rhythm. But an alarm clock, if it happens at the same time every day, will also entrain your body rhythms to some extent. And therefore, activity in your hippocampus will start to predict when that alarm is going to go off. So in a roundabout and indirect way, it will affect your hippocampus, not necessarily in a bad way by any means. But this is why, if you're having trouble maintaining regular sleep patterns, then that's why routine is so important. It just gives everything its best chance to line up with your innate biology, I guess.
We've had quite a few questions through about the quality of sleep. Sorry, I'm trying to find a specific one, but I'll sort of summarize across a few that have come through around the effect of using sleeping pills and whether the sleep induced through the use of drugs is actually quality sleep, and does it help? And over the longer term, are there risks over longer term use of sleeping drugs?
So that depends, of course. Not all drugs are equal, and no drug is perfect. So, benzodiazepines, for example, one of the earlier and most widely prescribed sleep drugs, put you to sleep in the sense that your head's on the pillow for longer, but actually impair brain activity during sleep. So these patterns of brain activity that are required to support memory processing are actually impaired by those drugs, and therefore they improve sleep quantity but degrade sleep quality. Now, since benzodiazepines, other drugs have hit the market, the so-called Z drugs, for example, like azopiclone, zolpidem. They are less damaging to patterns of brain activity, and therefore preferable if you want to conserve the contributions of sleep to your memory processing. And there are some studies that showing they can actually be beneficial as well. So in healthy sleepers, taking those drugs can improve memory to some small extent. So it doesn't make sense to be afraid of all drugs, but it does make sense to be cautious about all drugs. And the later generations of sleep drugs are certainly doing better than the earlier generations. But in many cases, we don't know the details of what these drugs do to sleep-dependent brain activity. So it's, that's just not looked at before they hit the market. And with hindsight, of course, it should have been. In the longer term, of course, there are risks. Again, the benzos, the risk of addiction. The later generations, that's less prevalent. But kind of in training your biology to a drug is always going to have a risk to some extent. Sometimes it is worth the risk, though, given how central having some sleep is to the quality of our lives.
Okay, thank you. And although we're on 8 o'clock, but there are two hopefully quick questions and sort of questions to round off on. And the first is, again, an amalgamation, although Beth asked here, how accurate and reliable are the sleep monitors on fitness trackers? But I think there have been questions as well from others about how can we accurately monitor the quality of our sleep? Are any of the things that are sort of available on the open market accurate and help us? Or do you become obsessed and fixated rather than actually really getting quality data about your own sleep?
Yeah, those are really timely questions because the monitor itself is out there, and at one point, one of the guys in my lab had an armful of fitness trackers to compare them all. Was also wearing the EEG, of course. It varies a lot. The average wrist-worn actigraphy and heart rate monitor tracker is reasonably good at telling when you fall asleep. It's reasonably good at detecting the non-REM deeper sleep stages, but it starts to get inaccurate, particularly in relation to REM sleep, which is hard to disambiguate from wake on the basis of the measures it's taking. And the lighter stages of sleep can be misinterpreted by the trackers sometimes as well. So I think they give you a kind of handy back of the envelope view of how much sleep you're getting, and to some extent its quality, particularly in relation to the deeper sleep, not so much for the other sleep stages. But that will depend on you. That will depend on your gender, your age, and lots of other factors as well. My 9-year-old wears a Fitbit, and it's not very accurate on him. Are they damaging in the sense that people start to obsess about sleep quality? Well, potentially, yes. But again, doing the cost benefit over the whole population, I'd rather that people paid well-informed attention to their sleep, and at the risk of some people becoming obsessed, than nobody paying any attention at all. Same as counting calories. On balance, it's good for the population to be aware how much they're eating, even if some get obsessed. So I certainly wouldn't advise against them on average.
Okay, thank you. And a question which I think has dropped off the pad, but I just really liked, that I thought we'd finish on, is, does cheese make you dream?
Me personally, no. And I'm digging back to my second year pharmacology lectures. I think in some people there may be some biochemistry behind that wives' tale, but it's the exception rather than the rule. So my advice is eat your cheese early in the day.
And if people take away anything from this lecture, then eat your cheese early in the day, I think.
Embrace the continental breakfast. Yeah.
Well, I think looking at the fact that the night is beginning to draw in, and it's clearly time for us to start to think about our sleep patterns and when we need to enter our deep sleep phase.
I can't reach the light switch from here, that's why.
I think all that really remains for me to say is just a massive thank you. Thank you so much, Matt. It's endlessly fascinating, the science of sleep, and I think we can tell from the sheer volume of questions that have come in beforehand and also through the question board this evening. I'm very sorry to everyone that we just couldn't quite answer as many questions as we had, but we tried to take a broad overview of the topics and the areas that people were asking. But Matt, thank you so much for your time this evening, for your leadership of neuroscience at the university, but for sharing such an insightful talk with us all this evening.
It's an absolute pleasure. If anyone wants to come and spend a night in the sleep lab, then just drop me a line.
Thank you ever so much. And so we're just about to wrap up, and Matt and I are off to put on our PJs and go to bed. However, at the point that we finish now, then we will open the chat room on here. And so that if you do want to stay on and want to chat to any of your fellow alumni on the call or on the event, then please do. One of the things that we were going to suggest that you could have a conversation about on the chat is if there are any particular topics or areas of research that you'd love to hear about from the university on a future digital event as well. So thank you ever so much for joining us this evening. We hope that it's been a really interesting evening. As I say, we will share a video of this. Thank you again to Matt, and look out, there will be an update very soon about our next event in around a month's time for our digital event series. So thank you for your continued enthusiasm for the university, and please do go and share the brilliant research that's going on at Bristol and enthuse. And when we share the video, share it wide and far with friends, family, and anybody else as well. So thank you ever so much. Do stay and participate in the chat room, but we look forward to seeing you at the next event. Thank you.
Sweet dreams, everyone.